Assalamualaikum and good day everyone. I'm Roswati Amin from University Malaysia Terengganu. I'm delighted to share with you our product, a tiny little crustacean harpacticoid copyboard, NiCoTox lacustris pacifica, as marine micromodel for ecotoxicity testing, also named as NiCoTox. Malaysia is one of the fast-growing nations in the global economy. In 2019, Malaysian economy ranked 27 out of 141 countries in the Global Competitive Index. Economic developments, however, has a certain environmental burden, including waste productions, oil pollutions, emissions of greenhouse gases from energy system, and deforestation. Malaysia is rich in water resources, but we are drowning our ecosystem in trash, oil, and carbon emissions. Ecotoxicity definitions characterize potential adverse effects from those activities which affect the organism through bioaccumulations and biomagnifications, and later disrupt the function of ecosystems, including human. Assessment of ecotoxicity of pollutant in terrestrial or aquatic environment to a certain organism is an essential component of ecotoxicology study. It is therefore necessary to find a suitable model organism that can help explain both acute and chronic marine pollutant toxicities with reproducible outcomes. Tests are commonly determined using fish, invertebrate, crustaceans, insects, birds, algae, amphibians, and rodents. In this project, we used NiCoTox lacustris pacifica harpacticoid copyboard, isolated from our coastal water Terengganu. This species is first recorded in Malaysia and now deposited at South China Sea Repository and Reference Center, UMT. Harpacticoid has been used extensively in ecotoxicological studies worldwide since 1970. This harpacticoid copyboard is able to tolerate a wide range of temperature and salinity, highly adaptability to culture conditions, and has been long considered as a good food source for fish culture. The relatively short life cycle, high reproductions, and population growth of harpacticoid copyboard make them ideal for use in experiments that rapidly assess the sublevel or chronic effect of water and sediment contaminant. The development of this species consists of 6 nucleal and 5 copepodite stages before they reach the adult stage. It requires only 9 to 12 days for the overall process. In terms of productions, the estimated cost is very minimal, with initial developments require only 150 ringgit Malaysia and 2 ringgit for monthly food maintenance. There is also a potential for commercialization with a price of 20 ringgit per 100 ml. The result of numerous environmental studies where toxicity tests have been applied normally are used to evaluate for ecological risk assessment. In our study, NiCoTox lacustris pacifica was used to evaluate the effect of synthetic and biodegradable microbeads on copyboard ingestion. Findings of this study already been published in high impact journal. Additionally, the result was to support the potential of PHA as eco-friendly microcleansing agent for commercial application. We successfully won several silver and gold medals in research innovation competition, including Grand Award winner in 2021. All of our findings were also delivered to the public through Marines Awareness Program at both primary and secondary school in Terengganu in 2019 and published in Utusan Malaysia in June last year. Move forward to the process. Coffee pot was first isolated from wild populations, differentiated between female and male, and then transferred to culture tank filled with filtered seawater. To maintain the culture, coffee pot were fed 3 times a week with a mixture of phytoplankton and bigger yeast. For monitoring the experiment, coffee pot were individually sorted and transferred to the desired concentration of substances or chemicals and incubated in situ parameter. Observations were conducted daily depending on the aims of this study. Other than research, this species has also been used as a model organism for teaching in planktonology subject under marine biology program. Analysis of different types of substances or parameters selected randomly by students have been applied in the laboratory to understand the potential effect of the selected parameter to the environment. Our result demonstrates the usefulness of this organism in future ecotoxicological study due to its important value in biological attributes as model organisms, the suitability for risk assessment to generate standardization and considering it for regulatory environmental agency in Malaysia. So that's all from us. With that, thank you very much. Assalamualaikum, salam sejahtera.